Dear students, today I will discuss on cell-mediated immunity. What is immune system? It is a complex network of different cells, organs and soluble proteins. An immune system protects us from foreign invaders. Now, immune system can be divided into innate immune system or response and adaptive immune response. Innate immune response is also called non-specific immune response and it is present in all forms of life. Whereas adaptive immune response is found in jawed vertebrates, that is vertebrates having jaw, and adaptive immune response has memory. Okay? Whereas innate immune response has no memory. Now, depending on the components taking part in an immune response, we can divide immune response into cell-mediated immune response and humoral immune response. Cell-mediated immune response is mediated by different cells like T lymphocytes, T cytotoxic lymphocytes, natural killer cells, macrophages, neutrophils, eosinophils, etc. Whereas humor means soluble. So, the immune system which is mediated by soluble protein or soluble molecules is called the humoral immune response. I will tell you about the cell mediated immune response. Now, all the cells present in our blood are generated in our bone marrow through a process called hematopoiesis. And hematopoiesis starts with a stem cell called hematopoietic stem cell. This hematopoietic stem cell is differentiated into two progenitor cells, lymphoid progenitor cell and myeloid progenitor cell. From myeloid progenitor cell, macrophages, neutrophils, these cells are generated. Whereas from lymphocyte progenitor cells, natural killer cells, some of the dendritic cells and T lymphocytes and B lymphocytes are generated. Now, T lymphocytes the initial part of the development of T lymphocytes takes place in the bone marrow. Then they migrate to the gland thymus and there they complete their maturation. That is why this group of lymphocytes is called T lymphocytes. Now, T lymphocytes come from the thymus to blood and B lymphocytes come from bone marrow to blood. And in blood, these lymphocytes migrate as recirculating or circulating lymphocytes, okay, and they are present at G0 stage of cell cycle. They don't divide until they encounter an antigen, and they are non-phagocytic in nature. If they don't encounter an antigen, they will die by apoptosis. But if a lymphocyte encounters an antigen, then it is called that lymphocyte is selected. It is called clonal selection. Okay, that particular lymphocyte will then start proliferation. It will produce many clones of its own. Okay, and they will differentiate into effector cells and memory cells. Effector cells show immediate effect against the foreign invader and memory cells keep the memory of that same foreign invader or antigen and it becomes active during the second exposure to the same antigen. Now, T lymphocytes can be of different types. T cytotoxic cells, T helper cells, T regulatory cells, T suppressor cells. Okay, and T cytotoxic cells are actually taking part in cell-mediated immunity. Now, T cells express a unique membrane-bound receptor on its surface. Okay, it is called T cell receptor. This is the structure of T cell receptor. We can see it is composed of two polypeptides. Now, it has a very short cytoplasmic domain which is unable to transduce a signal inside the T cell. That is why 3 pairs of heterodimers help the T cell receptor to transduce a signal. When it will transduce the signal? When a TCR will recognize a foreign peptide presented by another protein called MHC or major histocompatibility complex. Now, all nucleated cells of our body express a MHC molecule, major histocompatibility complex class 1, and 3 cells of our immune system, B lymphocytes, dendritic cells and macrophages. These 3 cells express a second type of MHC molecule called MHC class 2. So class 1 MHC molecules are found on all nucleated cells, whereas class 2 MHC molecules are found on the surface of antigen presenting cells. Now what are their functions? Class 1 MHC molecules present self peptides to the T cell, whereas class 2 MHC molecules present foreign peptides on their with them. Now T helper cells, the receptors present on the surface of a T helper cell recognizes foreign peptide presented by class 2 MHC molecule, whereas receptors present on a T cytotoxic cell recognizes non-self or altered self peptide presented by class 1 MHC molecule on any nucleated cell. So if a TCR, T cell receptor, recognizes non-self peptide presented by MHC molecule, then that T cell gets activated and it starts an immune response against that target cell, because something non-self presented means that cell has been infected with a foreign invader or our cell proteins has been changed. Now TC cells have no cytotoxic activity. The TC cell is called the naive TC cell. So cytotoxic T lymphocyte precursors are activated and differentiated into an effector cytotoxic T lymphocyte on encountering an antigen. Now how the activation takes place? For the activation, 3 signals are required. The first signal comes through the recognition of non-self peptide presented by MHC molecule on the target cell surface through the T cell receptor present on a TC cell. Clear? Second signal comes through the interaction between two proteins. One is present on the surface of the TC cell, one is present on the surface of the target cell. The protein which is present on the surface of TC cell is called CD28. It binds to its complementary binding site present on the B7 peptide or B7 protein which is present on the target cell. And the third signal comes through the interaction between a signal molecule called interleukin-2, which is a protein. It is secreted from the TH cell and it binds to its receptor present on the TC cell. And the third signal comes through the interaction between IL-2 and its receptor. IL-2 is called interleukin-2, which is a cytokine. That means it is a protein, signaling protein. It is secreted from T helper cell and it binds to its receptor present on the TC cell. Now when a TC cell or CTL precursor receives all these 3 signals, it is converted to functional CTL or effector CTL. Now it continues cell division and produces a clone of CTL of same kind. Okay, and some of them will be differentiated into effector CTL and some will be differentiated into memory CTL. Now the total process starting from CTLP to effector CTL, it takes almost 7 days. Now how CTL, effector CTLs, they kill the target cell. The target cell is showing non-self peptide via MHC class 1. The non-self peptide may come from a virus. Since viruses directly inject their genome, it is processed inside a cell, inside any cell, and when they are degraded, they are considered as self. And the non-self peptide may come from a transformed or cancerous cells also. Now, as a CTL recognizes non-self peptide presented by class 1 MHC molecule, it binds to the target cell more closely. Okay, and form a conjugate, and the conjugate lasts for 5 to 10 minutes. After then the CTL dissociates from the target cell and it goes on to bind to a second target cell, and the target cell after CTL dissociation takes only a few hours for undergoing apoptosis. So the target cell dies by apoptosis. Now what happens in the conjugate formation? TC cells or cytotoxic T cell precursors, they don't have any granules in their cytoplasm. But as it is converted or differentiated into an effector CTL, now many granules appear in the cytoplasm. And it has been seen that the granules rearrange or reorient themselves at the junction. Okay, and the granules fuse with the surface plasma membrane of the CTL, and they are released in the junctional space between the two cells. Now, what are there in the granules? Two types of molecules are present. 165 kilodalton A molecular weight protein called perforin, and a serine protease called granzyme B or fragmentins. Perforins, what is the role of perforins? Once the perforins are released at the junctional space, perforins are actually hydrophilic molecules, but as they touch the surface of the target cell, they undergo a transition, hydrophilic amphipathic or amphiphilic transition, and now they are able to penetrate into the lipid bilayer. Now, once a perforin gets incorporated into the lipid bilayer, many more perforins will come and attach to it, and they will form a hole, okay, having a diameter of almost 5 to 20 nanometer. And the granzyme B, which are also present at the junctional space, they will enter into the target cell through those pores, perforin created pores. And once granzymes are inside the target cell, it will start apoptosis. Another mechanism is there, which is called Fas Ligand binding. TC cells express a molecule called Fas Ligand, and some target cells express a molecule called Fas. When Fas Ligand binding takes place, it also sends a signal into the target cell to undergo apoptosis. Now, both the Fas Ligand interaction and granzyme B, release of granzyme B into the cytoplasm of target cell, activate the caspase mediated pathway.
Ma'am, what is caspase?
Caspases are proteins or proteases. They are called cysteine proteases. They are present in their inactive form in the cytoplasm of all cells. But once a caspase gets activated by a signal, okay, then that caspase cleaves other caspase molecules and make them active. And the cleavage takes place after an aspartic acid residue. That is why caspase got its name from cysteine and aspartic acid. So, both the Fas Ligand interaction and presence of granzyme B into the cytosol activates the caspase mediated pathway, and cells undergo programmed cell death, which is an orderly and systematic degradation of the cell. This way, CTL or cytotoxic T lymphocytes mediate killing of the target cell. Another important cell of cell-mediated immunity is natural killer cell. They are large granular lymphocytes, and they constitute almost 5 to 10% of circulating lymphocytes, and they provide protection or first line of defense against viral infection. As I mentioned, activation of CTL precursors to effector CTL takes almost 7 days. During that time, NK cells or natural killer cells, they check or prevent the viral proliferation. And these cells are also active against the tumor cells. They are cytotoxic and they kill almost all different tumor cells. Now, natural killer cells express a number of receptors on their surface. They don't have any receptor like the T lymphocyte or B lymphocyte, but they have a separate set of receptors on their surface. Two receptors are very important. One is called killer activation receptor or CAR, and the second one is called killer inhibitory receptor or KIR. Now, what is the role of CAR? CAR can bind to a specific molecule which is expressed when a cell is undergoing stress, like in case of transformation or cancerous cell. Okay, and when CAR gets its receptor and binds to it, it provides a killing signal, that is, kill the target cell. In case of human transformed cells, CAR binds to MIC-A and MIC-B, and when CAR gets the molecule present on a target cell, it provides the killing signal. But KIR looks for the class 1 MHC molecule, because expression of class 1 MHC molecules decreases in response to a virus inside the cell or in a transformed cell. So KIR looks for the class 1 MHC molecules. If it gets enough of class 1 MHC molecule, then it overrides the killing signal. The killing signal is overridden and the cell gets the survival signal. But if really class 1 MHC molecule expression has lowered, that means cell is undergoing some infections. The cell has been transformed or some virus has infected that cell, then it will be killed by the natural killer cell. Now the killing mechanism is same like the cytotoxic T lymphocyte killing. Since there are granular lymphocytes in the cytoplasm of natural killer cells, some granules are present constitutively. In case of cytotoxic T lymphocytes, they appear after the activation of that cell, but in natural killer cell they are constitutive and the granules are full of granzyme B and perforin molecules. So as soon as a natural killer cell encounters a target cell, it releases all the contents of those granules and the cell is killed. The cell undergoes apoptosis. Just the mechanism is similar to the CTL mediated killing. Another way of killing by CTL, and natural killer cells also secrete a number of cytokines, very important cytokines, and these cytokines activate the other cells of both the innate and adaptive immune response. Another way of killing by natural killer cell is mediated by antibody. It is called antibody dependent cell mediated cytotoxicity. What happens? Some tumor cells and virus infected cells, they express some proteins on their surface, and immune system recognizes those proteins and develop an antibody response. Now the antibodies developed against those proteins, they bind to the surface of the target cell. Now on the other hand, on the surface of natural killer cell, there are receptors for the Fc region of an immunoglobulin. Mainly CD16 is present on the NK cell surface, which can bind to the Fc region of an IgG molecule. So a target cell covered with antibody, now natural killer cells bind to the Fc regions and they mediate killing very quickly. Among the other cells which take part in the cell mediated immunity, macrophage and neutrophil, they are very important. Macrophages are developed from monocytes. Monocytes develop in the bone marrow through hematopoiesis. Monocytes come to blood from the bone marrow. When circulating monocytes, they migrate to different tissues, and in the tissues they mature into macrophages, and macrophages are named differently depending on the position of the macrophage. If they are present in the lungs, then they are called alveolar macrophage. In the liver, macrophages present in the liver are called Kupffer cells. Brain, the macrophages which are in brain called microglial cell, etc. And macrophages express a number of receptors on their surface. These receptors can recognize specific molecules present on the surface of an organism. The receptors are called pattern recognition receptor, PRR, and the molecules which are recognized by this PRR are called the pathogen associated molecular pattern. So as soon as PRR combines with PAMP, immediately phagocytosis takes place and the ingested organism is killed inside the macrophage. Macrophages also secrete a number of cytokines or small signaling proteins, and these proteins also activate other cells of the immune system. Another important cell is neutrophil. Neutrophils have granules inside it, and they are granular lymphocytes. And neutrophils are the first cells to reach a site of an infection or inflammation. So they are called inflammatory cells also. And neutrophils can phagocytose very quickly, and after phagocytosis, killing is very quick, much more than that of macrophages. Now how the phagocytosis takes place? As soon as an organism or a molecule binds to a specific receptor present on the neutrophil or macrophage, it is ingested, and as it moves inside the cell, then H+ ion concentration increases inside the vesicle. It is called a phagosome. That increased H+ ion concentration tries to denature the organism. Then it fuses with a lysosome. Lysosomes are full of different enzymes. Those enzymes act on the organism. Finally, they are destroyed. Okay, and the small parts are some parts are small peptides conjugate with MHC molecules, and they are presented on the surface. And rest of the degraded parts are released from the cell. Now, what happens inside the phagosome? As soon as a phagosome is formed, a membrane oxidase gets activated, and this oxidase now reduces oxygen to a superoxide radical. Superoxide radical accepts electron and protons, and it is converted to H2O2. H2O2 again accepts electron and proton, and it is converted to hydroxyl radical. This way, many different reactive oxygen intermediates are produced inside the phagosome, and in response to certain molecules like lipopolysaccharides, when a phagocytic cell encounters lipopolysaccharide present on an organism, or in case of mycobacteria, when the phagocytic cell encounters muramyl dipeptide, another enzyme called nitric oxide synthase gets activated, and nitric oxide synthase produces NO or nitric oxide, and this nitric oxide is very toxic for the microorganisms. So, both the reactive oxygen intermediates and reactive nitrogen intermediates, they kill the ingested organism. Okay, this way the phagocytic cells kill the foreign invader. Another important cell of cell-mediated immunity is called eosinophil. They are present at a very low percentage. Only 1 to 2% of eosinophils are found in normal individual serum, but their concentration increases in an allergic individual. Okay, because they mediate allergic responses. Now, what is the function of eosinophils? Eosinophils get activated in response to a parasite or its larvae. These cells are also phagocytic, and in the presence of parasites or its larvae, they phagocytose and try to remove the parasites. So, different cells of cell-mediated immunity have different mechanism of killing of the target cell or the foreign invader, but they work in combination. Each one of them tries to remove the foreign invader or the target cell infected by a virus or self-altered target cell or tumor cell, and they work together to remove that foreign invader.